So we all seen the handout for the spore stain?
Yeah.
Yes.
So again, we're going to talk about the process, the procedure, what it looks like. Then we have a few videos we're going to watch. We're also going to talk about negative staining, in particular a capsule stain. But first, the endospore stain. So what is the purpose of an endospore? What's its function?
Able to survive when conditions are not favorable, and then it could
Right,
produce.
so survival. Spores are about survival, right? And there were two major groups of spore-forming bacteria. What were those two major groups?
Bacillus and Clostridium.
Right, Bacillus and Clostridium. Remember, those are not the only spore-forming bacteria, but they're the two major groups. And they're both gram-positive rods. Now, when the bacteria sense that they're going to die soon, they'll start the process of sporulation to make a spore. So they replicate their DNA, and then they deposit layer upon layer of spore coat around that chromosome and form a very thick protective barrier. And then eventually the cell dies and releases that spore. While the spore is still inside the cell, we call it an endospore. But once the spore gets released, it's a free spore. But whether it's endo or free doesn't matter. We just look to see if the bacteria are making spores. Now, if the endospore is still inside the bacterial cell, it can be directly in the middle. We call that a central spore. It can be on the end, and we call that a terminal spore. And it can be somewhere in between the middle and the end, and we call that a subterminal spore. But we're not going to identify where the spore is. It's just that spores do not all form in the exact same place. It depends on what the organism is. But some spores are central, some are terminal, and some are subterminal. They're in between. We just said that Bacillus and Clostridium are the two major groups of spore-forming bacteria. But there are others like Sporosarsina, which is a coccus that makes spores, which is kind of unusual, but there are some other kinds of bacteria that make spores. But the largest groups are Bacillus and Clostridium. And every member of those species makes spores or has the ability to produce spores. So here we see that tough, thick spore coat, which helps to protect the chromosome inside. When favorable conditions return, the spore germinates and will form a brand new cell, which will then start to replicate and divide. So how do we do the spore stain? Well, our primary stain is malachite green. And so just like before, after you dry and heat fix your smear, you will go to a hot plate and put your smear on the hot plate, put a piece of filter paper on top and saturate the paper and the smear with malachite green, and you'll leave it there on the hot plate for at least 5 minutes. When you're finished, you remove the filter paper, put it in the trash, and then you decolorize the slide by simply rinsing it off with water. So water is the decolorizer for the spore stain. Rinse it off with water and then counterstain with safranin for about a minute and then blot dry. Now, just as with the acid-fast stain, the hot plate gets covered with stain, which means the bottom of your slide may be covered with stain. So here you are allowed to wipe the bottom of the slide. So you can use the 95% ethyl alcohol or the green lens cleaning solution and clean the bottom of the slide. right? Not the top, never wipe the top, but the bottom of the slide you can clean in case it gets all dirty. And then when you look at it, what you will see are cells that are stained red or pink. And we call these living cells vegetative cells. They're just ordinary cells. They're going about their business. And then we'll see spores. And they can be bright green, they can be dark green, and they can be kind of bluish green, like turquoise in color. But they are spores. In this case, we see a lot of free spores, right? They've been released from the cells. But like I said, whether they're still inside the cell or out, we don't really care. We're just trying to see if spores are present. Now, when you make your smears, although you make them the way you would normally make a smear, you touch and you look, it is okay if your smear is a bit thicker than normal. And the reason why is we really want to be able to find spores if they're there. If your smear is too thin, it may be really hard trying to find the spores. So with the spore stain, your smear, it's okay if it's a bit thicker than normal, which is what most of you would do anyway, because that's how you make your smears. But when it comes to the spore stain, a little bit thicker is fine, because that will give you more cells and more spores to be able to find. So that would be okay. Now the cultures that we would be given are either Bacillus subtilis or Bacillus cereus. So yes, we have to be serious in the microbiology lab. And the subtilis is subtle. So the B is subtle. It's not subtilis, it's subtilis, as in subtle. But they're both bacillus, so they both make spores. And then we would also use our soil plates. So remember, the very first day of lab, we did the environmental plates, and some of you would have tested soil. And we would have saved those plates. And we saved them for the spore stain because soil has a lot of species of bacillus in soil. And so we always have lots of spores in soil as well. So you would be able to see spores in one of the cultures that we make, as well as the soil culture that you guys had made. But the goal would just be able to find spores to be able to recognize them. So any questions on the technique so far? Nope. So we use the malachite green. We have to heat it up so it penetrates that thick spore coat. We decolorize with water because the malachite green doesn't really stick to anything else. And then we counterstain with safranin, and the vegetative cells are red and the spores are green. Now, remember when we talked about the gram stain, and where was the best place to obtain a sample for your gram stain? In the top half or the bottom half of the slant?
Bottom?
Right, it was the bottom half, right? Because the agar was thicker, there was more food, more moisture, so that's where you would find the younger, healthier cells at the bottom. But now you want to do a spore stain. Do you want young, healthy cells for a spore stain?
No, you would want the ones that are already going into that state. So they're
Right, so where would you find them on an agar slant?
probably
On the top?
Where?
The top?
In the top half, right? Because that's where the agar's thinner, right? The bacteria run out of food, they start drying up faster. So that's where spores are produced first in an agar slant, is in the top half. And it's especially true for a young culture. If you have a culture that's 24 to 36 hours old, you're probably not going to find any spores. But where they do start is at the very top. So something to think about if you were in lab, which you're not going to be, but if you had a culture and it's really young, you really need to go to the top to find spores. But if you have a really old culture, not only will you find spores everywhere on the slant, but near the top, it will be nothing but spores. So nothing but spores. All the vegetative cells are dead. It's nothing but spores. So let's say you were, let's say you forgot and you're making a gram stain and you grabbed your sample from the top of the slant and it's an old culture. When you stain it with a gram stain, you see a bunch of round or oval structures, and so you think you have a coccus, when in fact it's spores. So once again, it's important to always think about what you're doing as you're doing it. Where's the best place to get my sample, right? Depending upon what stain that I'm going to do. So for gram, you want to go to the bottom half. Spore, and you're at the top half, unless I tell you it's an old culture, spores are everywhere, right? If you go right at the top of an old culture, it's nothing but spores. That could potentially be confusing on an unknown if you were actually doing one and you took your sample from the top and gram stained it and saw nothing but little round things. And you might think you have a coccus, and that messes you up from the very beginning. So always try and keep in mind all these different things. Something else to remember. Let's say, you get your sample and you spore stain it and the entire cell is green. All the cells are green. Does that mean you have spores? If all the cells are green, is that positive for spores? If the whole cell is green, is that positive?
Yes. No, no, no.
Yes or no? If the whole cell is green, is that a spore?
No.
No, it's not, because the whole cell is not a spore, right? Spores are inside of cells, not the whole cell. So it's possible you're spore staining an organism which does not make spores, and yet the whole organism stains green. Can you think of another organism we've talked about that might pick up the malachite green and hold on to it, especially when you're heating it? Maybe something that has a thick waxy cell wall?
Yeah, was it the Mycobacterium?
Right, the Mycobacterium. So it's possible if you don't decolorize it well enough that your Mycobacterium will hold on to the malachite green. And so it looks like nothing but green. And again, that's a potential problem that some students have on the unknown where they do their gram stain and it's a gram-positive rod, and then they do a spore stain and everything's green. And they think it's positive for spores when in fact it's not. So again, if you were looking in a microscope, you'd be getting more and more practice, more and more experience, so you'd be able to recognize things. But there's a lot of stuff to keep in your head when you're in the microbiology lab. So that's what makes it a challenging lab, because you just have to remember so many things at the same time. So it is possible there are other organisms like Mycobacterium which may pick up and hold on to the malachite green. It's not common, but it can happen. But if the entire cell is green, that is not a spore, right? Spores are not the entire cell. So any questions on that? So we're going to watch a little video on spore staining. In this video, you'll see that they use a steaming hot water bath as a way to heat up the stain. We use the hot plate because this tends to be very messy. And you'll also see at the end when she adds the saffron in, she doesn't rinse it, she just blots it dry with the bibulous paper. But we would always rinse off our smears before we start blotting them.
I am now going to demonstrate the endospore stain. The endospore stain is a different type of differential stain. At the end of this stain, you will be at this point, the vegetative cells will appear pink and the endospores will appear green.
Start that over.
I am now going to demonstrate the endospore stain. The endospore stain is a different type of differential stain. At the end of this stain, you will be able to, through a microscope, differentiate between a vegetative or living cell and an endospore. Due to the nature of the endospore, this stain requires an additional step that we do not see in many of the other stains. This step is called steaming. To begin our staining process, we must begin with a smear that has been heat fixed. This was completed before the video began. The first step of the endospore stain requires the addition of the primary stain, malachite green. The second step of the endospore stain is a decolorization step. In this procedure, we will use water as our decolorizer instead of ethanol. Ethanol is too harsh to use for the endospore stain. Our third and final stain is the counter stain, saffron. To begin our procedure, I am going to get a small piece of bibulous paper and place this on top of the area that I have previously smeared and heat fixed. I then place the heat fixed slide and the bibulous paper on top of the steaming hot plate. The malachite green is applied to the paper and the slide while the steam is allowed to penetrate. This must be allowed to sit on the hot plate for 5 minutes. During the 5 minutes that this slide is on the hot plate, the steam will soften the walls of the endospore, allowing the malachite green to enter the spores. These cells will also turn green during this 5 minutes. Once the 5 minutes is up, while being very careful, you can take your slide off of a hot plate. I find the best procedure is to use the test tube tongs to slide the slide out of the steam and grab the slide. It is very important at this point to let your slide cool off before you grab the slide with your hand. Once your slide has cooled enough to touch, you then will remove your bibulous paper throw it in the garbage bin, not down the sink. That is the end of step 1 of the endospore stain. I am now going to apply my step 2 decolorizer, the water, in the same fashion that I did with my decolorizer in the gram stain. I will apply the decolorizer above the stained area, allowing the water to run down the slide. I allow this to happen until I no longer see any green running down my slide. I am now ready for my final step, the counter stain. I apply the saffron on top of my smear. This is allowed to sit for 1 minute. Once 1 minute has passed, now my vegetative cells would be pink. Shake off any excess, grab the bibulous paper to dry. My slide is now ready to be viewed under the microscope. At this point, the vegetative cells will appear pink and the endospores will appear green.
Are there any questions or comments about the spore stain? See, in labs, you guys would be having so much fun because your fingers are all green and red and they get very colorful. So why do we use the endospore stain?
To be able to see endospores.
Right, so the
Spores?
purpose is to find spores. And again, the malachite green is the traditional way to do spore staining. And it's still a very common way to do it, but as I mentioned last time, you can use the acid-fast stain to look for spores as well. But malachite green is the traditional staining technique. For what organisms do you expect to find spores? What are the two groups? Okay. What are the two groups? Spore-forming bacteria.
Bacillus and Clostridium.
Right, Bacillus and Clostridium. And once again, what's the function or purpose of a spore?
It's for survival mode.
Survival, right? So survival. So any more questions on the spore stain? Everybody's good with spores? Okay, next we're going to talk about negative staining, and in particular capsule stains. But remember that a negative stain is one that does not stick to the object or the structure you're trying to see. But since everything else is stained, including the glass slide, we can see that object or structure. And there are two types of negative staining that are commonly done. One uses Congo red and the other uses nigrosine, a black stain. And so we have several videos we're going to watch to see. But the red stain is often done as a capsule stain. And so remember, capsules are thick layers of polysaccharide. And we can see here that the slide background is stained. Even the cells are stained, but the capsules are not stained. As a result, we can see the capsules. I have some other examples. Again, the capsule stain, but a nigrosine stain. Here we see where the bacteria are very bright against a darker background. And here you can really see, right, the background is very, very dark with the stain. And here we see the bacteria, right? They're bright in color because they're not stained. And since this is bacillus, we can also see spores, little spores all over. So, but the negative stain, whatever you're trying to see, is not being stained. But everything else is, and that's why you can see it. So let's watch the first video, which does talk about using Congo red and also about capsules in general.
Structures known as capsules present outside their cell walls. Capsules are one type of bacterial glycocalyx. Capsules are structures composed of polysaccharides, proteins, or a combination of polysaccharide and protein. Bacterial capsules can offer the bacterial cells several forms of protection. Capsules can protect cells from desiccation and from host immune defenses. Pathogens that have capsules may prevent the ability of phagocytes to destroy them through a variety of mechanisms, and in some cases interfere with the activation of complement in the immune response. Capsules may also play a role in biofilm formation by allowing cells to stick to surfaces and to other bacterial cells. Biofilms are complex communities of microorganisms. Capsules serve as virulence factors for a number of human pathogens, including Streptococcus pneumoniae, Escherichia coli, and Klebsiella pneumoniae. In this lab session, you will be performing a capsule stain. The purpose of the capsule stain is to reveal the presence of bacterial capsules. The chemical composition of capsules causes them to repel most stains, so the capsule will not be visualized directly with a stain. Instead, the staining method relies upon revealing the capsule indirectly. The capsule is also a highly hydrated structure, in some cases consisting of greater than 95% water. Therefore, heating the sample will cause shrinkage and distortion of the capsule. So this staining protocol will be different than those you have already performed, because you will not heat fix the sample. You will follow the protocol for the capsule stain outlined in your NCTC lab manual. Please remember you will not heat fix your sample. The first staining solution used in your protocol contains Congo Red. Congo Red is a pH indicator that is red above pH 5.2 and blue at pH 3 and below. It acts as an acidic dye and is repelled by the bacterial cells, causing the background to be stained. You will place a drop of Congo Red solution on your microscope slide and then transfer your bacterial sample to the drop of Congo Red. You will mix the sample and then use a second microscope slide to spread the dye culture mixture into a thin film that forms a red background on the slide. The slide is air dried, but it is not heat fixed. After the slide is dried, a second staining solution containing Mannavol stain is added to the slide. Mannavol stain is a mixture of acetic acid and acid fuchsin. The acid fuchsin is a basic dye that stains the bacterial cell red. The acetic acid in the stain lowers the pH of the sample on the slide, causing the Congo Red to appear blue. Capsules do not interact with either dye and therefore remain unstained. Upon observation, you will see a grayish-blue background with red cells. If a capsule is present, it will appear as a white halo around the cell. Provided on the screen are the results that you should expect in the laboratory for the 5 species that we have been working with. Do not expect to find a capsule for the Mycobacterium spigmatis. For all other samples, a capsule should be present. The image on the top right-hand side shows what a capsule would look like in the laboratory setting. The bottom image shows an organism lacking a capsule.
So that's one. Now we'll look at the nigrosine stain, which is a black dye. Using India ink.
Hi students, this is Professor Schimmel and Renee, and we're going to show you today how to do how to make a negative stain. We do negative staining to detect the presence or the absence of capsules on bacteria. And here's how we're going to do it. We're going to start with a nicely scrubbed down microscope slide. Use a pipette to put a couple of drops of water at one end of the slide. Then we will aseptically remove a little bit of the organism from the slant that Renee prepared, and that organism will get mixed into the drop of water. Now, we're not going to spread it out at this point. We're just going to mix the organism into the drop of water. All right, after we do that, we're going to add 1 drop of India ink to the water and organism mixture. Now, please take care not to touch the tip of the bottle to the water, or you're going to contaminate the entire bottle of India ink. All right, once we've done that, we're going to pick up a second microscope slide, and we're going to use it to drag that material across the entire surface of the slide. All right, now once you've done that, you've got live microorganism on the end of that slide. So what you're going to do is you're going to give it a rinse with alcohol, and then wash it, and you can use it from there. We don't happen to have any alcohol with us, so we're just going to set it down in the sink for now. Okay, now we're going to let this slide air dry thoroughly. We will not heat fix, and we won't put the slide on the slide drying rack either, because it's just going to get too warm. If we do that, or if we heat fix the slide, even if there are capsules present, they're going to just shrivel up from the heat. So we're going to let that negative stain air dry completely. Then we are going to take the slide and we're going to put it on a staining rack over the sink. And we're going to cover it with safranin. We'll leave the safranin on for a minute. Then we'll pick the slides up one at a time, give them a quick rinse. You can use your clothespin for that, or just do it by hand like Renee is doing. Give the slides a quick rinse, blot them dry with your bibulous paper, and you're ready to view. And I will add some photos of what capsule slides look like at the end of this video. All right, you guys, thanks for watching.
Any questions or comments?
So you don't use heat for this one?
No, because the heat is going to deform the capsule. It's going to cause it to shrink and to deform. So no heat fixing, and sometimes not even heating on the slide warming tray. So it takes time for the smear with the ink on it to dry. So now we're going to see another version. Or not. Yep. My computer froze up for a second. We have to wait. Let's see. Okay, now it'll work.
In this video, we're going to look at the negative stain technique. Now, the reason we use the negative stain technique is because it gives us a slide where the bacterial cell is very clear and distinct. Now, you'll recall when you do a bacterial smear, you have to heat fix the slide. Anytime you use heat, it distorts the cells. With the negative stain technique, we don't use heat. So the cells come out more natural, you get a much better image. Now, the reason we call it the negative stain technique is because we're applying a stain that carries a negative charge. Now, you'll recall that bacterial cells have a negative charge too. So the cells actually repel the stain. So we're really staining the background. So what you get is a white cell against a dark background. You get really nice contrast. So let's look at the materials you're going to need to do this technique. You'll need a bottle of negatively charged stain. I'm using nigrosin. You'll need a bacterial culture, an inoculating loop, a staining tray, 2 microscope slides, a heat source to sterilize your loop, and finally a slide warmer. So let's look at the technique itself. First, what you want to do is take one of your slides and put a small drop of nigrosin near one end. Now, don't get it on your fingers, so I go about, probably a ½ inch from the end of the slide, and I put a little drop of nigrosin. Next, we're going to get a culture of bacteria and we're going to stir them into the drop of dye. Let me flame my loop, sterilize it. I'll let this cool. I'll go in and get me a nice sample of bacteria. And I'm going to stir these into the dye drop. But when I stir it into the dye drop, I'm just going to stir it side to side. So I'm moving the drop laterally as I'm mixing the bacteria into it. And stay away from your finger. You don't want to get the bacteria all over you. Now immediately flame and cool your loop to destroy the excess bacteria. And now you're ready to spread out the dye. So in this part, you take your other slide, your spreader slide, and you put it on your base slide at about a 90-degree angle like this. Then lean it towards the dye drop, towards your fingers, and pull it up so you're pulling it into the dye drop. And the dye will grab onto the slide. And then you push it away from your fingers, and that will spread the dye out. You just keep spreading it out, and then as you get towards the end of the slide, just roll the spreader slide off. Now this is contaminated with bacteria. So you want to immediately drop this into a biohazard bin or into a beaker full of bleach and water. Now we're going to take this and we're going to put it on our slide warmer. Now as the water evaporates, the dye actually bonds the bacterial cells to the slide. And you end up with your finished product. Now let me put this on this bibulous paper so you can see what the final product should look like. There you have it. Now a few things you want to avoid. Make sure that when you do this technique, you do not put too much nitrogen on the slide because otherwise you'll be waiting forever for it to evaporate. Also, make sure you don't get the slide on your fingers because it's loaded with bacteria. So that's pretty much the technique, and let me show you an image of what the bacterial cells will look like in a negative stain technique.
So any questions or comments? Nothing? So did you notice there were, slight variations in technique? Not everyone necessarily does the staining procedures the exact same way, right? Because there are, variations in terms of how you can do some of these procedures, and they, give you the same kinds of results. So no questions or comments?
Can I ask him a question about the material on the test?
Not just yet.
Okay.
Because I want to stop the lab recording to do that. So today we've gone through the endospore stain and the negative stain. On Wednesday, we're going to talk about motility and how that works, and also just talk about micropipetters, which I don't know if you were taking 150 in the spring, you may not have gotten to use them before we shut down. So we will talk about micropipetters just because we would use them, and so you should be familiar with them, even if you can't actually, handle them and use them. So, anything else? About lab. You guys need to talk more.